Hello, everyone. On behalf of the editorial office of BMEH, welcome to today's webinar. We're dedicated to have you all here. To ensure small session, we kindly ask everyone to keep your microphone muted during the presentation. Today's session will begin with the talk from Professor Jesus Santamaria, followed by the discussion session led by Professor Filippo Rossi. Now it's my pleasure to hand over to our host, Dr. Guotai Li from University of Healthy and Rehabilitation Sciences. Dr. Li, the floor is yours.
Okay. Can you hear me? Okay. Thanks very much for the introduction from Jenny. I'm Guotai. I will host the seminar in the following time. Today we invited Professor Hans Santamaria to give us talk. Now let me introduce Professor Santamaria. Professor Santamaria is a professor of, I think it's the next slide, Jenny. Yeah. Dr. Hans Santamaria is a professor of chemical engineering and Pharma chair of nanomedicine at the University of Zaragoza in Spain, as well as group leader in the Nanoscience and Material Institute of Oregon. Professor Santamaria is among the leading European research on the science of nanomaterial and application in the field from unconventional catalytic to nanomedicine. He has obtained numerous projects with European funding, including ERC advances. He has published 385 peer-reviewed manuscripts and have received over 22,000 citations. He has also received numerous awards, including the Millennium Research and Future Award, the prize for excellence in research awarded by the government of Oregon. He is the academic in the Academia Europe, and the Institute Lambrando Academia de Science in Latin. After the report of the Professor Santamaria, it will be the time of discussion. So we will also invite an expert participating into the discussion, Professor Filippo Rossi. So the next slide. Yeah. So I will also give an introduction of Professor Rossi. Dr. Rossi is an associate professor in the applied physical chemistry at the Department of Chemistry, Materials and Chemical Engineering at Politecnico di Milano from Italy. He received a PhD in Industrial Chemistry, Chemical Engineering from the same university. He has a lot of research experience abroad as a visiting PhD student at Imperial College London and as a postdoctor at Uppsala University and as a visiting professor at Tokyo University of Agriculture and Technology, and the University of North California, and so on. His main research interests as the field of innovative polymeric material for nanomedicine, drug delivery, and tissue engineering with experimental and modeling studies. So now we are going to start the presentation. Professor Santamaria, I will hand it to you. So, Professor Santamaria, can you share your slide?
Okay. Yep, thank you. I'm going to share the slides.
Yeah.
Okay, can you see my presentation now?
Sorry, I can't.
Okay.
Now we can.
I can.
Now we can see it.
Okay. Okay, very good. Well, thank you. Good morning, everyone, and many thanks for the invitation to this forum. I'm very happy to share our research with you and hope for a stimulating discussion after this 45 to 50 minutes presentation. So I would like to discuss catalytic nanoparticles in the context of cancer treatment and also exosomes as the delivery vectors for these catalytic nanoparticles. So in the talk, we will first give some motivation why we need new paths against cancer and why selective drug delivery is still the standing problem here. Then we will discuss briefly those problems and why catalysts can be the response, the answer to that. Then why this is not being yet implemented? So we will discuss two problems that have hindered this implementation so far and the solutions. Then some very encouraging results in recent years from our lab and some conclusions. Okay, so I will think that cancer may be the big failure of science in the sense that we are looking at 30 million new cases and 13 million deaths in the world for 2030. That's more than 30% increase from 2020, where we had 10 million deaths. And this is, affecting 20% of men and 1 in 6 women in cancer in their lifetime. In spite of many therapies like the traditional chemotherapy, radiotherapy, and surgery, but then emerging immunotherapy, targeted therapy, hormone therapy, hyperthermia, etc. So the problem with chemotherapy is, of course, that we are not selective. I mean, killing cancer cells is not too difficult, but we are not able to deliver chemotherapy drugs to cancer cells alone. And this is giving rise to a lot of many different effects on the body, which are sometimes very devastating on many organs and tissues of our body. Some effects will quickly disappear, but many take months of years. And in fact, secondary effects is what is mainly limiting the amount of chemotherapy that the doctor can give. So the dose problem is the central problem here. I mean, if we don't have the dose, when you look at cancer cells, chemotherapy is designed to kill them with a sufficient high dose. But at these high doses, already normal cells are being affected. And since when we receive chemotherapy in systemic administration, the drug travels throughout our organism, it will affect many other types of cells. So what we think is that we need to develop therapies that do not get consumed like old chemotherapy drugs, and instead can prolong their action. That could be, for instance, hyperthermia enabling nanoparticles. In that case, we can go to optical hyperthermia, where we have a near-infrared particle that in the infrared we have a higher penetration of the light, and this will heat up the cells. Or we can use magnetic hyperthermia that in principle doesn't have any penetration limits. So this is one possibility to have a therapy that can be with the same particles in the tumor can be done again and again. But it is also possible to use catalytic therapies. And this is also very, very interesting because in that case, the catalysts do not get consumed. So catalysis against cancer is an idea that has been being used for now about 10 years. Before that, it had been left out of cancer therapies, but catalysts are in principle ideal tools for that because they can facilitate chemical reactions in a complex environment such as the tumor microenvironment. And also, it's an ideal tool to solve the dose problem. In the dose problem means that if we don't have enough dose reaching the tumor, the cancer cell will survive and develop treatment resistances. But with a catalyst, the dose problem is much better because even if a small amount of catalyst reaches the tumor, as long as it is active, it will produce therapy inside the tumor. So nanometric catalysts are ideally suited for this task. It can be nanoparticles or it can be enzymes. We have been working on both. We will concentrate this talk on heterogeneous catalytic nanoparticles. So the concept here is that you consider the tumor as a chemical reactor where reactants are consumed and new products are made, and the products are mainly cancer cells that will stay in the tumor. And we want to put catalysts inside the tumor to change the chemistry of the tumor towards a scenario that will stop tumor growth. So we want to make the toxic molecules inside the tumor instead of delivering them throughout our body and hope that they will reach the tumor. think about this. This is a blood vessel, and this is a growing tumor. And when a tumor grows, a very strong alteration with uncontrollable cell replication will happen. And this will require a steady nutrient supply. So there will be nutrients here coming together with oxygen. So the first thing that the tumor will do will be to recruit new blood vessels. So this is a sign that the tumor is growing because it will need a lot of nutrients. Because of the tumor growth, there will be parts of the tumor that will be hypoxic, so there will be a very strong drop of oxygen concentration. There will be metabolic alterations with give us a lot of opportunities because these metabolic alterations take place in cancer cells. For instance, we know that the tumors will have a high nutrient demand to sustain anabolic energy needs. In the tumor energy cycle, we go through glycolysis rather than through the conventional Krebs cycle, meaning that we going to have a very large consumption of glucose, okay? In spite of the fact that in aerobic glycolysis, only 4 moles of ATP are made per mole of glucose instead of 36 moles in phosphorylation. So this gives us a first opportunity. We have glucose to target. And also because of this high lactate production, the pH in the tumor will be lower. So that's altered metabolism. Because of this altered metabolism, there will be also a lot of reactive oxygen species production, such as hydrogen peroxide, superoxide, or OH radicals. So this means that the tumor to survive will produce a lot of antioxidant molecules. So GSH is the main one, glutathione, and they will be overexpressed in tumor. All these characteristics give us the main features of our reactor. We have a reactor that will work at 37 degrees. It's a chemical reactor where you cannot change the temperature. They will use a lot of glucose. There will be a lot of glutathione around, very low oxygen concentration, mildly acidic pH, and a lot of hydrogen peroxide. This is the main features of our reactors in the tumor. And we need to find suitable catalysts and reactions for this environment. They need to be able to work under these conditions. And we also want catalysts that can be selectively activated in the tumor microenvironment because we don't want the catalyst to actually damage other cells. So we want to, if possible, activate them. Okay, so for local activation of the catalyst, we can rely on ambient activation by conditions that happens in the tumor, or remote activation. In that case, we are going to use mainly near-infrared light activation. What are our catalytic strategies? We have catalysts that can do one of 3 things. One of the first strategies is to deplete key molecules for the tumor growth, and this is called starvation therapy. So we are going to target glucose, glutathione, and amino acids. There are other possible objectives, but I will be presenting you the results on these 3. We can also enhance the natural processes for the generation of reactive oxygen species. So there is already many of those, but we can produce more and produce an imbalance in the tumor so that the tumor will not be able to counteract it. And finally, there is another possibility that we can use things that are not in the tumor microenvironment at all. In this case, these things are already in the tumor. These things are already in the tumor. But in that case, we were going to introduce completely different molecules that are not there, and we will convert a prodrug into a drug inside the tumor. So these are the 3 strategies that we are going to pursue. For strategies 1 and 2, we're going to combine them and deplete key molecules and at the same time generate reactive oxygen species. This was our first proof of concept. It was published 8 years ago. And we use gold titanium nanorods as near-infrared activated glucose oxidase depleting catalyst, okay? That will deplete glucose and generate reactive oxygen species. This is our catalyst. It's a gold nanorod coated with a titanium structure. And this gold nanorod is sensitive to near-infrared light. So when it receives near-infrared light, it will inject electrons on the titanium coating around it. This is just the characterization of the catalyst, and this is the absorption. You can see that there is a very strong absorption at 800 nanometers. That's near infrared. We coated with titania. It's still there, a little bit of displaced, but we have a lot of absorption inside the in the near infrared. It works very well in the reduction of glucose when it has the coating. If it doesn't have the coating, it doesn't work. So we're producing gluconic acid, mannonic acid, and arabinonic acid, but mainly gluconic for the oxidation of glucose. At the same time, we deplete the glucose, but we generate hydrogen peroxide. We see it by this probe reaction. We can also see with probes that we could generate superoxide and OH radicals only when the catalyst is coated with titanium. If not, the gold alone is not able to do that. So in that case, it's interesting because we have a catalyst that can deplete glucose and generate reactive oxygen species, and it's activated by near-infrared light. So it means that you can put a laser into the tumor and activate it only there. Second example I want to give you is simultaneous glucose and glutathione depletion plus reactive oxygen species generation. Look, this is the natural process taking place in the tumor. Okay, the tumor has an enzyme that is expressed there is the glutathione peroxidase. So a molecule of glutathione will use one molecule of hydrogen peroxide, will deplete one molecule of hydrogen peroxide. It's a selenium-containing hormone, and it will actually consume glutathione to deplete hydrogen peroxide. The glutathione gets oxidized to the dimeric form. Okay, so this is what the tumors are doing to counteract the high concentration of hydrogen peroxide. We want to do the same with a catalyst. We want to do the same with a catalyst that can work in a redox mode, take a molecule of hydrogen peroxide and produce OH radicals, which are worse than hydrogen peroxide, and at the same time gets oxidized. We did that with these particles. They are spinel structure with a cubic copper iron structure. The main interesting thing of this catalyst is that it's very selective to the presence of glutathione. When there is a glutathione present in the system, the amount of copper leached from the particle, it increases by more than 2.5 times, okay, compared to the iron. The iron is much less expelled. And you can see, when you do a mapping, you can see indeed that the copper is being depleted while the iron is still there, okay, after use. Okay, so this catalyst is able to release copper. And when it releases copper, we don't have time to go into the details of the mechanism, but you can read the mechanism there. The copper release will complex with a molecule of glutathione, oxidize to the dimeric form, get oxidized there, and then go back to copper 2 by using oxygen from the reaction environment to produce hydrogen peroxide and superoxide. Now, this is very interesting. Well, by the way, we were able to demonstrate this mechanism, but this is interesting because we see how fast the GSH is being depleted. So it's very, very effective in depleting glutathione. It's also very effective in producing superoxide and radicals. We can see that. And these superoxide radicals they can do many things, but one of the things that they can do is to deplete glucose. So you can see here that when you have, in the presence of hydrogen peroxide, the glucose, when the hydrogen peroxide is added, it gets depleted very fast. So it works by depleting glutathione and creating superoxide anions. But of course, the main problem is that you need oxygen for that. So that when it comes the other part of the catalyst, the part that doesn't get leached is the iron-rich part that will act as a phenton-like catalyst, taking hydrogen peroxide and producing oxygen. And we see that every time we add hydrogen peroxide, we get an increase in the oxygen. In that way, we alleviate the hypoxic nature of the tumor because we need this oxygen to generate, to deplete the hydrogen peroxide and generate the superoxide level. Okay, when we did those results were in test tubes. Now we did the same in cells. So here is you have two type of cancer cells, this glioblastoma and cervical cancer, and a healthy cell, mesenchymal stem cell. And this is the level of glutathione before put our catalyst. So you can see the intercellular glutathione in that case is about 5 millimolar. In that case, it's about 6 millimolar. We did that by HPLC. And in the case of a healthy cell, it's much, much lower, about 5 times lower. It's only about 1 millimolar. This is very nice because in the absence of glutathione, the catalyst don't get activated. So after using the catalyst, you see the glutathione levels go down in the cancer cells very strongly, okay, very significantly. But in the case of the healthy cell, it doesn't go down. So the catalyst gets activated mainly in cancer cells because of the high glutathione content. So when we do more examples with other type of cells, you can see that the 50% toxic threshold is about 1 order of magnitude lower for cancer cells than for healthy cells. So this is nice because it can activate selectively by the ambient, can activate the Okay, the next example is an oxygen-free reaction. In that case, we found by there was previous results by the group of Strasbourg that copper could also do transamination. Okay, in the case of transamination, we found that when you have an amino group that is close to an alpha carboxylic acid, that's in a peptide or in an amino acid, in the presence of pyruvate that is in all our molecules, the copper ions in the system are able to produce the transamination. This amino group moves here, moves to here to produce alanine. So this is transaminated and the corresponding keto acid. So we checked that, and it worked very well with glutamine, glutamic acid, and aspartic acid. And we see indeed that inside the cells, if you don't put any glutamine in the system, there is a natural consumption of glutamine. But when there is copper catalyst releasing, you have a lot of stronger reduction that basically completely depletes the glutamine inside the cells in less than 72 hours. The catalyst can also do GSH, glutathione, and the dimeric form transamination because they also have an amino group next to a carboxylic group here. So what we see when we do the same is that not only transaminate amino acids and take away amino acids from the tumor, but also takes away glutathione and especially the oxidized form because the catalyst is more fast to oxidize it, but then takes the oxidized form and depletes it, constantly producing aniline. So alanine. So this is a very interesting reaction because it doesn't need oxygen. It can take place without any oxygen in the medium. It is only a transamination. Okay, that's another case of ambient activation in the presence of glutathione. The catalytic strategy 3 involves prodrug activation. In that case, the idea is to use something that is not toxic and then use a catalyst to make it toxic, to take away a group there. So we do this in collaboration with the University of Edinburgh, the group of City Brosetta. And the idea is that we will put something that is not toxic in the body and will only be converted into a toxic form inside the tumor where the catalyst is. So the rest of the body will not have a toxic concentration, but inside the tumor they will be. Okay, let me give you just one very, very recent example. We just published this very recently. In this case, we have platinum nanodendrites, that is a heterogeneous catalyst, about 25, 30 nanometers in size. We first tested it with a dye. So we have this dye that is green fluorescent, and we inactivate it by putting a chemical group there. We tried these 4 different chemical groups, and we found that the one that worked best is this one. So we have this with this group is not fluorescent, And now the catalyst is going to take away this part and make it fluorescent again. Okay, so we did the experiment of injecting it in vivo in zebrafish by putting cancer cells inside the pleural sac of the zebrafish that already these cancer cells already contain the catalyst, but just the cancer cell and the catalyst. The catalyst will not kill the tumor by itself, is not toxic. And now we put this fish to swim in a medium where the product, the molecule with this, is there. So the fish is swimming in the product. The product that gets inside the fish will get converted. And this is exactly what happens. If there is no catalyst, there is no fluorescence. If there is catalyst, you see a lot of fluorescence being converted inside the fish. Now we do the same with a prodrug, a cancer prodrug. So now you have this cancer prodrug that is inactive. Okay, we with the catalyst, we are going to take away this part, all this group, and make it active again. This is SN38 that is highly cytotoxic. So you can see that it works very well in the tumor. It will reduce the tumor. With the platinum, it doesn't get reduced. With the prodrug, it doesn't get reduced a little bit, not much. But with the prodrug and the catalyst, we do this cleaving. Okay, so this can work very well. But remember, we are not doing selective delivery here. We are just injecting things inside the fish. It can work, just to show that it can work in vivo. Okay, so the take-home message is that we have very effective catalysts for tumor therapy with selective activation by near-infrared or by the tumor microenvironment condition. But what about the delivery? We need to take this catalyst to the tumor. So the first problem was to get catalyst, and the second problem is to actually do selective catalyst delivery to the tumor. So we need to develop new ways of selectively delivering our catalyst to the tumor. The tip is that this is an old problem. It's the same problem in nanotechnology that we have been trying to solve for 30 years. How to deliver nanoparticle to tumor. So let's go very, very briefly back to the methods that is being used to deliver nanoparticle to tumors. So the first thing that is being used, and this is what is at the heart of all approved nanomedicine so far for cancer, is the passive delivery, the so-called EPR effect, standing by enhanced permeation and rotation effect. It was discovered by Masumura and Maeda in Japan in the 80s, and they demonstrated that in the tumor, there is a lack of lymphatic drainage, and there is a lot of penetration because the tumor grows so rapidly that the blood vessels are defective. So there are penetration between 50 and 400 nanometers. And if you put nanoparticles there or proteins, they will be preferentially deposited there. So this is the basis of the system, the leaky blood vessels and the poor lymphatic drainage due to the fast growth of the tumor. So if we can produce particles that will circulate for a long time, chances are that they will concentrate in the tumor. So you need to retard the detection by the immune system, and to do that, typically you will put around your nanoparticle, you will put polyethylene glycol or something similar to increase the circulating time. This is EPR effect is what is being used in the cancer medicines approved so far, like Doxil, for instance, or Abraxane. Okay, the second possibility is let's not rely on passive delivery. Let's put active delivery so we can decorate our nanoparticle with targeted ligands, such as antibodies or peptides. And then these ligands will recognize epitopes in the cancer cell and stay there. So there are many things that have been tried in the last 3 decades with a lot of targeting options, very sophisticated. I'm just giving you 2 reviews from 10 years ago to show that we already have everything there. I mean, we have dendrimers, antibodies, peptides, aptamers, you name it. There's already many things decorating the part. Okay, So we have passive delivery, we have active delivery, and now the bad news. In 2016, this paper by William and co-workers in Nature Review Materials was very depressing to read because it showed us that doesn't matter what you do, if it is inorganic material or if it is organic material, and if the targeting active strategy is passive or active, we are looking at less than 1% of nanoparticles in the tumor. Okay, on average, they showed that looking at the literature, the average amount of particles that go into the tumor was 0.7%. Okay, and it was again tested many times, and it was demonstrated that this is true. This is consistent. This is the reason why the nanomedicines tested in clinical trials, more than 200, mostly fail at phase 2 and 3 due to poor efficacy. And the reason is that they don't get to the tumor. Okay. So what can we do? Because the problem cannot be solved by passive targeting, we just saw it, and the problem cannot be solved by the active targeting, because there is in 2018, this paper that was very, very interesting because they put nanoparticles coated with active targeting trastuzumab or folic acid, and only 0.7% of these actively targeted nanoparticles reach the tumor. So active targeting is also not good. So what are the options left to deliver therapeutic nanoparticles, including catalysts to tumor cells? We are betting in my laboratory for now a few years. We are betting on Trojan horse strategies. In this type of, you remember the story of Trojan horse, the Greeks put a lot of warriors inside, and then the Trojans allowed it into the city. And the idea is that we put something that the tumor will not recognize as hostile. So we want to use tumor homing cells like stem cells or dendritic cells, or, and this is what we are going to do, vesicles, typically exosomes. So exosomes are something known since the 80s also. This is the definition given in 1981, vesicles derived from the exfoliation of the plasmatic membrane. All of our cells basically can produce exosomes, so they will constantly produce exosomes and expel them. They are present in most biological fluids, and early studies thought that they were the garbage of cells. They thought that the cells will use exosomes to release things that they did not need. But today we know that this is not true. Exosomes are key elements for communication between cells, and inside the exosome there are lots of proteins, amino acids, and biomolecules that are essential for many biological functions. So this is like an exosome, how they look like. This is taken in our lab in electron microscope. You can see that they're between 50 and 150 nanometers, and they have this characteristic structure of the membrane, this double membrane. So this is the size distribution. And why we are interested in them? We are very interested because of the selective targeting capabilities. Because exosomes come, the membrane of the exosome comes from the membrane of a cell. It is claimed that exosomes can reach these same cells, okay? They are able to do a multiple recognition event and go preferentially to their progenitors. So that means that we take cancer cells or cells that home into cancer cells and use their exosomes and load them with our nanoparticles, maybe they will have a much better selectivity. Okay, so you don't want to touch the membrane. The membrane is very important. And the problem is, of course, how to load your nanoparticles into exosomes. So I give you two examples. We were loading 40 nanometers hollow gold nanoparticles into exosomes. These are hyperthermia nanoparticles. And this is a relatively large size. So we tried a lot of things. We tried incubation at room temperature, incubation with saponin, thermal shock, okay, freeze thaw shocks, thermal shocks different at lower temperature. We tried sonication and we tried electroporation. And what we found is that to actually get these nanoparticles inside the exosomes, we have to use very strong conditions. And in that case, the exosome membrane will be mostly damaged. This is what the picture shows. And instead, what we did was to incubate our nanoparticles with the cells and wait until the cells expel them. So in that case, we had exosomes containing nanoparticles, and the membranes were not damaged. So this is less efficient because it depends on the cell. So some exosomes are loaded with 4 nanoparticles, some exosomes are loaded with just 1, some exosomes are empty, so you really don't have so much control. But the good news is that you can get exosomes that are almost intact. And I will show you in a minute some examples of how these exosomes are very useful. The other possibility that we have is when we have very small nanoparticles. So, like in this case, we have platinum nanoparticles. And in that case, we can load them by using a very mild sonication. And you can see that mostly the exosomes are intact. Or we can also use very mild heat shock. And in that case, you can see that we get exosomes loaded with platinum nanoparticles. And they seem to be mostly intact. And indeed, we use them for cancer treatment, and we do get a very significant reduction of the tumor growth by these exosomes containing platinum. Okay, this is still not published. Okay, the other possibility that we have to load our particles is to use the ship in the bottle approach. So we harvest exosomes typically by ultracentrifugation. We put them into contact with a precursor of the metal, like a chloroplatinic, chloroauric precursor molecule, and then we use just carbon monoxide in the gas phase in an autoclave to actually reduce it. Why we use a carbon monoxide reduction? Because the carbon monoxide is a gas and doesn't leave any liquid residue. So after that, you get a very controllable production of exosomes inside of nanoparticles inside the exosome. And I show you here some examples of palladium nanoslabs loaded in the exosomes by this method. Okay, so this is the other possibility to actually load novel metals into exosomes. Okay, so I'll show you some in vitro selective tropism of exosomes loaded, in that case, with gold nanoparticles. These are the gold nanoparticles that I'll show you that were loaded by the natural procedure, just allowing them to get internalized. So here you can see I will show you the video, but I want to tell you what you are going to see first. These big cells are mesenchymal stem cells, okay? And you can see that they are full of particles. They are gold particles. What we see is not the particles themselves, but they are agglomerates, okay? So we see them as black dots. And here, the small cells are monocytes. The monocytes are scavengers, so you're going to see them moving around. So what we did was to track the exchange between cells, and you can see here that there's one cell that has lost this, and it goes into the other cell. The monocytes are there. They look at it, but they don't eat it because they recognize, probably, this is what we think, they probably recognize that it's coated with a membrane that is not from their species. So we have dozens of videos like this. In all cases, it starts from a mesenchymal stem cells, goes to another mesenchymal stem cells, and the monocytes go around without touching it. That's in vitro, of course. So the next thing that we did was to co-cultivate mesenchymal stem cells and cancer cells, allow them to exchange between them particles for a long time. And since these particles are hyperthermia particles, then we put a laser there. And what we find is that we kill selectively only the mesenchymal stem cells that already had their particles inside. So even though we allow them a very long time for exchange, we are not killing these cells, we are only killing the cells that have. Again, showing that the interchange is very selective. In vitro, okay, this is in vitro. The next thing that we wanted to know is that if our catalyst inside the exosomes was going to be active as a catalyst and selective, at least in cells. So this is what we did. We took a cell culture, we isolated the exosomes. So this is the exosomes. Next thing that we do is to put them into contact with a palladium, a precursor, put them in under the carbon monoxide atmosphere, and we select, we generate these nanoslabs, palladium nanoslabs. They are very, very thin, like 4 nanometers, mostly inside the exosomes. This is a cryo, and this is a conventional high resolution TN. So you can see them in both cases. And we see when we do protein analysis, we see that the main exosome markers are still there. Okay, so we have an exosome that is loaded with palladium nanoslabs, probably with catalytic activity, and keeps the markers characteristics of exosomes. So next thing, we want to try to make this catalysis here. This is the prodrug, this is the inactive form, okay? So what we are going to do is to take away all this part here with the catalyst and then make it into active. Okay, so this is the panovinostat. It's a relatively new drug that does this, is cytotoxic. Okay, so there is a very strong difference in toxicity between the 4th and the 5th, okay? So this is the cytotoxic, and it's 21 times more toxic than the inactive form. It gets very well internalized into cancer cells. That's not a problem. So now this is the interesting part. Look, we have 2 different types of cancer cells, okay? So A459 and U87, they are both cancer cells. We put them in contact with the palladium exosomes. They are not killed, okay? When we put them in contact with the inactive form, this form, they are not killed. When we put them in contact with the active form, both cells are killed. I mean, there's more than 80% killing in both cases. And when we put them with the inactive form and with a catalyst with exosomes of this A549 cell, when we put the U87 cell, it doesn't get killed at all because the catalyst is inside the exosomes of the other cell and do not get internalized. Instead, when we put them in contact with the catalyst with their own exosomes, there is a lot of killing, more than 50% killing, because the prodrug is being transformed into cytotoxic. So the delivery works very well in vitro with the catalysts. What about in vivo? Okay, in vivo we tried first use exosomes from mesenchymal stem cells because they also have tumor tropism. We load them with hyperthermia nanoparticles. So we wanted to know if there is an improvement in vivo against EPR effect, and also if exosomes loaded with nanoparticles were able to find disseminated tumors. So this is our particle. This is the hyperthermia absorption. You can see that they are very able to heat up the water. This is in a test tube. And this is the exosomes loaded with the nanoparticle. You can see that they are still there, the markers. Okay, so next thing we do is to go to a xenograft. That's the xenograft with the tumor. We are going to inject our nanoparticles inside the tail vein, and then we will put a laser on the outside. So what you see, basically, the luminescent data are better, but I just want to show you the real data because the luminescent data have some artifacts. You can see that there is a lot of particles inside the tumor. This is postmortem. And the important thing is that this is the EPR effect. 0.4 of the gold gets to the tumor when you just use the nanoparticles coated with polyethylene glycol. When you put them inside exosomes, you multiply this by 4 almost, okay? And that's not a lot, okay? It's only 1.5%, but it's 4 times more than the EPR effect. And when we do metastatic tumors, like we put a disseminated tumors in the pancreas liver region. You can see the tumor luminescent is all the way there. We do the same. We see the particles inside the tumor when we inject them in the tail vein. And in that case, we increase from 0.7 for the pegylated nanoparticles to 1.7. So it's less than it's 2.5 times in this case. How good are they at reducing the tumor growth? But for the xenograft model, after signing the laser, you can see that there is very, very, very strong reduction. Sorry. And for the multinodular model, also, we get the same reduction, okay? Even though it's only 2.5 to 4 times better than the EPR effect, is enough to produce the reduction of the tumor. We really see the tumor disappear from at least from luminescent data. And we see a lot of reduction in the Ka67 proliferation mark. Okay. In vivo, we also did the palladium exosome treat the chemistry, the catalytic chemistry, in this case with this inactive prodrug to some inactivated form of paclitaxel. So same thing, we injected in the tail vein of the tumor. And what we saw in that case for the palladium nanoparticles is that when you use pegylation, more than 90%, 95% go to the liver. In that case, less than 70% is in the liver. We see them in the tumor after 48 hours or 1 week. So we can see them in the tumor only about 1% when it is EPR effect, about 3%, and then 2% still after 1 week when we put them inside exosomes. And this is again enough to produce a very strong reduction of tumor. Finally, my last example is a delivery in vivo of 2 nanometer particles in the exosome. So same thing, we use in this case the carbon monoxide procedure to load the exosomes. You see that there is we compare them, same amount of platinum, the exosomes with platinum compared to cisplatin, that, as you know, is a standard drug for cancer treatment. So we injected free cisplatin, okay? And what you see here is that it's a very strong reduction in the liver, and very strong, even though it's small. And we still have in the liver, with the exosome, we have in the liver a lot of platinum, but in the tumor, you see that almost 3 times higher when we put the platinum inside the exosome. Again, we see a very strong reduction of the tumor with the exosomes. But what is most important is that we had to stop, for humanitarian reasons, we had to stop the treatment with cisplatin, because at this concentration, the weight of the mice stopped. They basically stopped eating. Their digestive system stopped functioning because of the high toxicity of cisplatin. Okay, so we have a much lower toxicity alternative with platinum. Okay, this is my last slide. The conclusions are that new approaches are certainly needed in cancer nanomedicine. We strongly believe that heterogeneous catalysts can play a new role in oncology by modifying the tumor microenvironment, depleting key molecules in situ, creating reactive oxygen species, and converting prodrugs into toxic products. And we have some examples of remote and local activation. And we see that Trojan horse strategies have exciting advantages for targeted delivery, but we need to improve them a lot. Thank you very much for your attention. I just wanted to thank the people who actually did the work, my co-workers here, my colleagues in Edinburgh and Instituto de Salud Carlos III, and thank you all for your attention. Thank you very much.
Okay, thank you too, Professor Santamaria. Thanks very much for your wonderful presentation. Now we are going to...
Okay.
So yes. You also have a slide of the acknowledge, sorry.
Sorry?
I saw you still have a slide of knowledge.
Oh, yes. Yes.
Do you
So
want to?
if we need it, we can go back to the slides, but I think it's better to stop sharing for now. Okay, you agree?
Okay.